Hi, my name is Michele Goodfellow, and I'm a graduate student at the University of West Florida in the Department of Earth and Environmental Sciences. So I have a background in forest resources and conservation from the University of Florida. And fortunately, I've been able to use my background in studying trees in order to apply it towards this degree program here at the University of West Florida. We're standing right now in Joe's Branch, which is a tributary of Olive Creek. And the purpose of this project is to study the impact of stream restoration on the downstream areas, the different tributaries in this watershed. Olive Creek watershed is a highly urbanized area. We're located in southeastern Alabama, right between the cities of Spanish Fort and Daphne. And so this is right before the I-10 bridge over to Mobile. So there's a lot of different infrastructure in the area, and in fact, the amount of non-porous surfaces, meaning like driveways, parking lots, is about 40 to 50% in this area. So there's a lot of urbanization. So what I'm studying, as you can see here in the stream bank, is the riparian forest directly behind me. So I look at the impact of stream restoration on riparian forests. The goal of this project is to determine if stream restoration further upstream and other areas of this urbanized environment are going to impact the way that the stream moves down, the way that the stream moves and migrates, and the way that the vegetation grows. A healthier habitat generally is going to have less invasive species, and they're going to have more of the natural vegetation that you see behind me. So trying to quantify those changes takes a little bit of field work. So that's why we're standing in a stream today. One of the things that we do is called a Riffle Cross-Section. So we're standing in the riffle of a stream, which is a shallow part of fast-moving water. And the reason that we're studying this area is because it's an area where there's higher species diversity and different species in a riffle than in a deeper, slower moving part of the river. So the way that we study this is we look at changes in topography. So this right here is a transect line that we've laid out. It's measured in meters, and it goes back on 15 meters back from each river bank or each stream bank. And the reason we go back that far is because we want to quantify the floodplain where that riparian forest is located. So we measure this area using one of these. This is a stadiarod, which generally speaking is just a very large meter stick. So we use this meter stick along this transect at every 0.5 meters. So we'll put it at every 0.5 meters. And then we'll take a reading from this using our survey level. It's basically a large scope that's on a tripod that allows us to see these numbers from a far distance. What I'm going to do with this data eventually, after I study the streams for about a 2-year study period, so I'll come back every 6 months and redo the topography along with the other items that I'm studying. And I'm going to compare those topography changes from the 6 months, or from every 6 months over that 2-year period, to determine if there's fluctuations that are related or correlated to upstream restoration taking place. So not only am I looking at the topography in the floodplains, but I'm also going to be looking at vegetation. As I mentioned earlier, non-natural vegetation is a sign of degradation. It's a sign of human impact. It's a sign of your overall decreasing habitat quality. So we quantify that by doing overstory vegetation or vegetation plots. And so we use something known as a wedge prism, which is just a calibrated piece of glass, in order to determine our plot size. And then we measure that for diameter at breast height and to determine the structural layer. So trees at different heights are a different part of the canopy. The highest part is what we'd consider your canopy. The low part is our ground cover. And then in between, we could have an understory and a midstory. Having a larger diversity of that structural layer provides more habitat for different animals. So if we have more structure or more structural diversity, then we potentially have more habitat, or we have more habitat potential in the forest. So we want to quantify that. In addition, your ground cover makes a large difference on the habitat available. Animals need a source of food. So if you have deer, for example, they might need some forbs to munch on. If you have birds around, you want some trees that might produce some seeds or some berries. So you want to make sure that you have some live vegetation in your understory where that food source is generally coming from. So we quantify the understory much the same way on this transect. And then I'll go through every 0.5 meters along the transect and look at what is intersecting on the ground. So if there's ground, bare soil, we'll quantify that. Live vegetation. Leaf litter. Now leaf litter is important because that's a sign of decomposition. Generally, when you have decomposition, you're going to have macroinvertebrates and other insects and bacteria that are in the area. So this right here is an automatic level or an engineer's level placed on top of a tripod. And we use this, it's basically a large binocular or scope. It just allows us to magnify the view from this end, or from the viewfinder end. And so we use this to read the numbers on the stadia rod so that we can determine those changes in topography. Because we're looking down 30 meters from this part to the other side of the stream bank, on the other side of the stream. So the way that this works, you look through the eyepiece, just like so. You want to make sure that the area is perfectly, or that the level is perfectly level in order to ensure that you're having an accurate reading. And so you'll focus in on the stadia rod and take your measurement.